pathophysiology of ovaries, largely with regards to the various hormonal mechanisms, how the ovaries develop, what are the various regulatory factors which are there, and how do we have a normal cycle. So gonadal development basically is essentially in the form of a double model. So as we have discussed about, then whenever we talk about various endocrine nodules, we have got the pituitary, where you have got the adenohypophysis, which is a glandular structure, and a neurohypophysis or the posterior pituitary, which is a neural structure. When you talk about pancreas, you have got the exocrine and the endocrine part. When you talk about thyroid, you have got the follicular and the parafollicular. Again, neuroendocrine versus glandular. So all the usual endocrine structures, adrenal, cortex and medulla, they are all glandular and neural origin. Similarly, gonads have a dual origin. So the main structure of the gonad is actually formed in the urogenital ridge, while you have got the germ cells which come from the hindgut. From there, they migrate from there to the gonads. So 4 to 6 weeks of life, you have the urogenital ridge which develops. And from the urogenital ridge, you have got 3 major structures which develop. And they include, Dhwani? So renal, adrenal, and the gonad. So all are common. So you have abnormalities in one, you will also have abnormalities in the other structure as well. So now germ cells are coming from outside, and we'll talk about how they migrate and how their maturation happens from that perspective, around 6 to 7 weeks. Now on that, you've got multiple various genes which start working in the form of a transcription factors, so which result in development of the bipotential gonad. So the gonad is structurally same. There is no difference between testis and ovary. So this is more likely to be a bipotential gonad. And from there, you have got multiple genes which are involved in the development of the adrenals. So you've got the NR5A1 which develop the adrenal, the WT1 which develop into the kidneys. So if you have abnormalities in WT1, you will have renal as well as gonadal abnormalities, and you may develop nephrotic syndrome, mesangial sclerosis, and Fraser syndrome and Dennis-Drath syndrome, which can develop from that regards. Now, once the bipotential gonad is developed, the WT1 and NR5A1 will then result in development of the gonad and the expression of a very, very important determinant of the direction of gender development happens, which is SRY. So SRY, we all know, is the sex determining region in the Y chromosome. In the presence of SRY, largely NR5A1 and SRY will then result in activation of the SOX9, which result in development of the Sertoli cell. And this Sertoli cell will then start developing the various aspects of production of AMH. You will have also the Leydig cell production, which will happen, and then from there the testicular development happen. So testicular development requires all of this. You need GATA4, you need CBX2, you need WT1, you need NR5A1, you need SRY, you need SOX9. So it's a very, very demanding thing from there. And then, of course, you need to have enzymes to produce testosterone. You need enzyme to convert testosterone into DHT, and you need to have an androgen receptor. So all this is essential for the male development, so to speak, from that perspective. Now, this is a process which we'll discuss much more when we talk about testis in that regards, how NR5A1, SRY play a major role. Now, ovarian development few decades ago was considered to be a passive process. So you do not have a SRY, automatically you have a default mode of development of ovaries. But now what has been shown that there are multiple genes which are involved in ovarian development. Some of them are fundamental, like the beta-catenin pathway, which is basically involved in the cartilage development, in the ovarian development, the bone development as well. And you have got specific ones which are also acting as an anti-testis genes, like FOXL2, which causes granulosa cell growth, but also inhibits the Leydig cell and Sertoli cell, and DAX1, which is again known as the anti-testis gene. So there is an ongoing fight. It's not just SRY which determines. So there's an ongoing fight which is there between SOX9 and FOXL2 in deciding. So if FOXL2 is more, it will become like an ovary. If it's more like a SOX9, it will become like a testis. That's why if you talk about a XX male, so what will be the most common cause of XX male? XX male means complete male. So that will be the SRY insertion. So if you have SRY, you will become like a male even though you are XX. But these male are not fertile because you need so many more genes to really have fertility. Now, if I go the other way around, Vibha, what are the causes of XY females? I'm not talking about serogenic defects and other things in terms of a genetic, complete XY females.
It could be right from the beginning. If there is a SRY delay, there is an absence of SRY, there is a SOX9 relation, there is NR5. In fact, adrenal will be also associated. So
XX male, most of them will have SRY insertion because it's very, very difficult for a male to have unless you have got the SRY. But for a XY female, you can have hundreds of problems. You can have no bipotential gonad. You may have no WT1. You may have no NR5A1. You may have absence of SRY, SOX9. All of them will ultimately result as a XY female. So XY DSD, quote unquote, or most severe form XY female will have much more causes. Similarly, if you have DAX1 overexpression, if you have a FOXL2 overexpression also, it will bring it down. Even though SRY is there, you will not have development of that. So the other thing to remember is that suppose you have a defect in any of this pathway, what will be the phenotypical presentation in an individual at birth? So if you have a defect in any of these pathways, right from NR5A1, so they will have a normal female development. This is very important to understand. So to have a XXDSD, you need to have an active process. It's an act of commission. You need more androgens coming in. While to have a XYDSD, it's an act of omission. No gonad, no testis, no testosterone, no DHT, no androgen receptor. Yes, it's a XYDSD female development. But for a female to have a XX to have a male development, you need to have a SRY. You need to have a testis. You need to have an adrenal producing your androgens, like in the setting of CAH. So XXDSD is an act of commission. XYDSD is more of an act of omission from that perspective, and that's important. So there are various pathways which become important. So now we have talked about how the bipotential gonad develops and how the ovaries start to develop. Now from there is the process of proper ovarian development, which requires multiple timed transcription factors. Now, what are these transcription factors? So if you remember pituitary, I talked about there are various factors which act at different ages of gestation. So you will have the proximal ones like the LHX3, LHX4, HESX1, which are involved in the hypothalamic development. Then you get the prop1, which starts the pituitary development, the pit1. Same ovarian development also has a synchronized. So they are like orchestras. One will start, other will stop. So you have got the very basic ones, which are the mesenchymal basically genes, WNT4 and RSPO1, which is fundamental for ovarian development. WT1 and R5A1, of course, is involved in the bipotential gonad. If you don't have a bipotential gonad, you will not have ovary. The problem is that even if you don't have ovary, you will have a normal female development. These girls will present with primary amenorrhea. They will not present to you basically in the initial phase from that perspective. Now, beta-catenin is a very, very important pathway. And then you have got the secondary ovarian developing genes, of which BMP2, which is a bone morphogenic protein, plays an important role in ovarian development along with DAX1. And then from there in ovaries, we know that you've got the theca cell, which basically produce the androgens. Theca cells are producing the androstenedione, which is important from that perspective, and they will also be producing progesterone. The oocyte, which will basically be involved in terms of the reproductive functions, and then you have got the specific pointer, the figula and nobox, which control the granulosa cells using the GATA pathway, which produces estrogen and AMH. So estrogen and AMH are the major products of the granulosa cells. So if you have the FSH level in a male who has hypergonadotropic hypogonadism, that means he has got a testicular failure, would their FSH be higher as compared to the FSH in females with hypergonadotropic hypogonadism? Klinefelter versus Turner's, whose FSH are higher? You've seen reports of Klinefelters and Turners. Vibha. Why? Because FSH is suppressed via estrogen as well as inhibin, and testis produces much more inhibin compared to ovaries. So that's why the FSH levels are much higher in females with gonadal failure compared to males with gonadal failure. That is something which is important to understand from that regard. And of course, you've got the myoid cell. Now, if you now look at in terms of the germ cells, so germ cells, I've talked mainly about the ovarian stromal development. The germ cells around the same time. So 4 weeks, I talked you have the development of the urogenital ridge. 6 to 7 weeks, you start the bipotential gonad developing. Now, 6 weeks in the hind gut, suddenly different types of cells start to emerge. These are the germ cells. And this is very fascinating. So you don't have the same area where the germ cells are produced. They are coming from a very far off place. When I talk about GnRH, I'll also talk about how GnRH neuron migrate from the olfactory placoid to the hypothalamus. So there's a very risky scenario. If they don't migrate, then you don't have any reproduction. So these germ cells will then migrate using a chemotactic process, a long path for them. So there are inhibitory pathways like TGF-beta, which will attract them. And this process will result in the germ cells to get lodged in the ovaries. Now remember, we talked about bipotential gonad. The germ cells are also bipotential. So there is no difference between a oocyte and a spermatocyte in the beginning. What determines that this germ cell will become a oocyte or a spermatocyte is the environment. It's the effect of environment. So conditions which are favoring meiosis will develop into a oocyte. Conditions which are not favoring meiosis will result in mitosis, and that will go towards a spermatocyte. So the main difference is how your ovarian environment and the testicular involvement is, which one is favoring meiosis. That's what it decides from there. So once the germ cells go there, there is STRA8, which specifically says, okay, I want more meiosis. And only those germ cells which are entering into meiosis will be saved, all else will be killed. We'll talk about this, how their numbers of follicles and they change, but only the meiotic cells in the ovary will survive. Otherwise, you will have no survival. And there also are no box figula, we already discussed, are their part. This will happen by around 10 to 11 weeks. So it's pretty early in terms of gestation. Very importantly, once the meiosis happens, it is stopped at the diplotin stage. It's not that the meiosis will continue. It's just like a commitment. So it's like you can say that you have got engaged. So they have got engaged to the ovary, so you will stay there, but they are not going to get matured here. It will take time. So this will happen. When is the further process of meiosis happening? So this meiotic arrest is happening fetally. Now, when would be the next level of meiosis happening? During puberty, and final would be when actually the sperm is going to enter, there will be the trigger which will be there. So this is a meiotic arrest which happens in this stage, and again bone morphogenic protein becomes important. Now this is a bit complicated but very, very important to understand. So when we talk about oocyte, it's a single cell. Now oocyte first of all has to go for meiosis, otherwise the ovaries will destroy it. Next step is that it has to be surrounded by cells to protect it. So it has to be strong enough. So now the whole game is to select the most favorable oocyte. Now, what are the difference between this evolutionary selection for sperms versus ovaries?
Like this covering and all?
No. How do the body selects the best sperms and the best oocytes? How is this process different?
So the sperms that are more motile, that will be.
So it's a race. Basically, you've got millions of sperms, and the one which are racing, the most agile, the most rapid will be able to reach. But here the process is a bit different. So you will start off first, you're doing meiosis or not. I'll select you. If you're doing meiosis, whether you are good enough to save yourself around making this wall. And then the body will say, okay, I will recruit 300, 400. And then gradually from that, you have got the follicles will start to emerge. And then the most dominant follicle will be taken. Others will be destroyed. So this is again an evolutionary process to get the most agile and the most stable oocyte, which will be taken up for further requirement and development from that regards. So you've got oocyte, then primordial follicle is just surrounded by a single layer. Then you have a primary follicle in which this layer becomes from the squamous epithelium to columnar epithelium. Then the follicle grows, and then finally you develop a cavity inside, which is known as the antral follicle, and then ovulation, which we'll talk about a bit later. Now, different stages are regulated via different genes / hormones. Now, remember that figula we talked about very early, GDF9, BMP I'm talking many times, TGF beta, these are very important for the early development. Then comes an important player which is known as the anti-Mullerian hormone. So we know anti-Mullerian hormone causes suppression of the Mullerian structure. But at a lower level, so normally at around 20 or so, you will have destruction of the Mullerian structure where it is coming from the testis. At lower level, it will cause small follicular growth. So follicle will grow from this pathway, and then you got other genes which will basically ensure that you have a growth process which happens, and then KIT ligand comes into the picture. All of these are genetic / paracrine. So what I'm trying to say is that the development of small follicle is independent of the hormone. Why does it need to be independent also? Because when we talk about the ovary, they are in a sheath. So these oocytes do not have a blood flow. So they can't get the hormonal signals to begin with. So initial development is completely independent of the hormonal status. And then from that perspective, you start to have the FSH effect at a later stage. Now, what is the name FSH? It is follicle stimulating hormone. So the main work of FSH is to stimulate the follicle. But what FSH does is that it will now push AMH out of the system as well. Because AMH, what it is doing, it is that it is okay, allowing initial growth, but it doesn't allow beyond a particular level. And then FSH comes into the picture, it says, okay, it's like a maturity. So you can think of it that somebody is in a primary school. So you've got a teacher who is actually teaching them till primary school. Beyond that, it is not allowing too much growth, because if there is a too much growth, then the oocyte may be disrupted or if it gets too much knowledge. But after a level, there will be a need for further knowledge. So then FSH comes in, the first one goes off and FSH starts to go. So if you have high levels of AMH, what will happen basically is that you will have many small follicles. And AMH is being produced by small follicles. So small follicles produce more AMH, produce more small follicles. So what happens in PCOS? You have got, essentially you don't have a dominant follicle. You've got many small follicles. They produce more AMH. So high AMH in the blood. Now remember, all this is paracrine. So when we say, okay, blood levels may be a good marker, we are not really true in that perspective. But if you have too high levels, the blood levels will go up. So blood level is not the best marker of what is happening inside the ovary. But yes, if your AMH is double the normal range, it means you've got many small follicles happening. And people often say, okay, AMH is a very important investigation in PCOS. No. AMH is just telling you that if it's high, that there may be a PCO appearance. So it's a surrogate marker of PCO appearance. That's it. So what I'm trying to say is that very early development is largely genetic or environmental or paracrine, which is AMH. After that, you have got the FSH coming into the picture, which happens at a late stage. Now, from there, FSH increases the size of the follicle to a big extent. You have got IGF-1 coming into the picture. And this is very important because in insulin resistance, the IGF-1 levels become high. Insulin is high, IGF-BP level goes down, free IGF-1 is more. So you will have more ovarian stromal growth. More ovarian growth means more androgen. So insulin resistance causes hyperandrogenism. So this is when I talk about PCOS, the same process will come into the picture. And SMAT pathway, which you were discussing about in that NUPL2, also becomes important. Now, after the FSH has really made the, so now this follicle has gone through the primary school, the secondary school, it is now the test phase. When it is entering, there comes LH. So LH, you can say, is like a tertiary level teacher. And it's basically going to stimulate it too much. And after that, ovulation will take place. So LH will come into that picture. And estradiol also when the levels are very high. When we talk about the follicular phase and the luteal phase, you will know that the initial estrogen is very low. So it's not going to do much benefit. By around 10-12 days, the estrogen level goes up, and then it will have an impact on the ovarian development. So now if you talk about, you can classify the ovarian development into two parts. One is non-endocrine, which is still primary follicle or early follicle, and then endocrine. So now based upon that, Alapan, can you tell me what will happen if you have somebody who has got a FSH receptor problem, somebody who has got a LH receptor problem, or somebody who has got a aromatase problem or a estrogen receptor problem in terms of ovarian morphology?
Since problem is there, multiple small follicles will be present, but no dominant follicle.
So what is happening is that AMH is working a lot. It is causing a chaos. So it's like that if you have got a strong central leadership, then the regional people will become less. If you have got a weak center, so FSH is not there, all the regional satraps will start going up and they will cause chaos, and there will be a lot of PCOS-like appearance. Yes. So FSH defects will cause problems in terms of very smallish multiple follicles. What about LH? Yes. So if you have a LH problem, the problem will be ovulation will not happen. And that is the main problem. If you now contrast this to boys, LH is more important in boys compared to FSH. LH deficiency means you can actually have a DHD, LHCG receptor defect, or you have a hypogonadotropic hypogonadism. FSH defect will not cause a problem in terms of normal development. The problem will be infertility. In girls, LH defect will not have a problem in terms of pubertal development because some LH is there. It's enough to produce androgens. Local androgen production is there. So your estrogen production is not affected, but your ovulation will be affected. While FSH defect will cause complete problems in terms of amenorrhea, there will be not enough estrogen. Plus, you will have follicles which are multiple small. Now, finally, what about estrogen receptor defect like ER alpha? We discussed on that day. It will be late follicular. So you'll have a large follicle. So large ovarian cyst will happen in that. So morphology can be determined where your problem is. Then you have got a development of inhibin. That's a late stage. Inhibin basically will tell FSH, okay, now my follicle is much bigger. I don't want your levels to be very high. Come down, and that will have an important role. And we'll discuss these individually as well. And activin is opposite of inhibin. And you've got follistatin, so all three will cause a modulation. So most important from my perspective is late growth is FSH. Before that, AMH will become important. Now, what do androgens do to ovarian morphology?
So, this androgen, the small follicles will cause increase in the.
So what does androgen do to this?
Testosterone will also suppress the estrogen.
So what is the major effect of testosterone in the ovarian, this process of oogenesis, I would say?
It is converted by aromatase.
No. What is its effect? High androgen, what will it do? Main effect on this process is that dominant follicle will not get formed. So if you have high androgen, dominant follicle will not be formed. What it means is small follicles will continue to grow, and small follicles produce androgen. So hyperandrogenism will cause multiple small follicles. Those follicles will produce more androgens, and this will be a vicious cycle. So what I'm trying to say is that the polycystic ovarian appearance. Now, what's polycystic ovarian appearance? You don't have a dominant follicle, you've got multiple small follicles. It is the effect of hyperandrogenism. It may be a contributory factor, but it's not the cause of hyperandrogenism. So hyperandrogenism causes PCOS. PCOS appearance alone is not the cause of hyperandrogenism. That's what you need to understand. Now, of course, you said that it has an effect on the progesterone inhibition of LH. If you have got hyperandrogenism, progesterone does not inhibit LH, so your LH goes up. Your LH goes up, you will have a high LH-FSH ratio, your testosterone levels go up again. So a lot of people say high LH-FSH ratio is a marker of PCOS. No, it's an effect of hyperandrogenism. Anybody who has hyperandrogenism will have a high AMH, will have a polycystic ovarian appearance, will have a high level of LH and FSH. And this is not the cause, it's a contributory factor, but the primary pathology is somewhere else. So if you have CAH, if you have adrenal tumor, all of them will have a PCO appearance. So just you have a PCO appearance doesn't mean that this is PCOD. This is something important to remember, and androgen disrupts the development of the dominant follicle. Now, how do the other structures develop? So this is the internal development, and we know we've got the Mullerian duct, the Wolffian duct, which basically are going to be, if you don't have enough testosterone, the Wolffian duct will go off. And because there is no AMH, your Mullerian structures will start developing. So what really are the Mullerian structures, Alapan?
Which structures?
Yeah, which structures develop from the Mullerian duct?
That like ovary, fallopian tube.
Ovary?
Fallopian tube. Just the upper part of the vagina is one.
Okay, so upper third of the vagina and uterus and fallopian tube will develop from that regard. So ovarian development, of course, is a distinct phenomenon. The lower third of the vagina is a urogenital sinus. So whenever you have got somebody who has got a Mullerian agenesis, basically your lower third of the vagina will be there, but it will be a very small vagina. As you had seen in that case, that you don't have very much, it won't be allowing, it will be a blind vagina from that perspective. So in the absence of AMH and in the presence of estrogen, Mullerian duct will develop into the fallopian tube, in the uterus and upper part of the vagina. This is how the structure will develop. Of course, if there is a XY individual, they will produce AMH. AMH will cause suppression. Wolffian duct will then start developing into the other structure and Mullerian duct will go off. What about external development? So again, external development is quote unquote passive. So you can say that if there is no hormones, you will have this development. So you have no androgen, then you will have a normal development of the labia majora, labia minora, and the other regards, but estrogen will play an important role. One point which is important to remember is that 8 to 12 weeks are the most crucial phase in terms of sexual development, at least externally. So in a XY individual, in a male, the fusion will happen by 12 weeks. In a XX individual, fusion will happen only till 12 weeks. So if you have a male who has got a deficiency of testosterone after 12 weeks, quote unquote, hypogonadotropic hypogonadism, because HCG is taking care of their testosterone production, they will have micropenis but no hypospadias. Similarly, if you have an excess of androgens after 12 weeks of life in a XX individual, you will not have labioscrotal fusion, you will have clitoromegaly only, which will happen. So this